Okay, here's the last figure that we're going to talk about. Again, figure in chapter 20 and then the associated text in section talking about cloning. And cloning is making a identical twin. And the way that's done is we've learned that, well, we should be able to take any one of your cells and it will have, with rare exception, it has the whole genome. It has the entire genome you got from your mom and an entire genome from your dad. It's a diploid cell. It's got one copy from mom, one copy from dad, and it's got the whole genome. And so in theory, any kind of like more or less any one of your cells should be able to make a whole you, an identical twin. And why can't it do that? What can we do to learn how to do that? So that's what this figure is about, and this idea of cloning. And it fits into this again. We've been talking about this final theme. Fits into this final theme. The concept of totipotency is this idea that the cell has all the information to make a whole organism, and it's all about regulating a subset of gene expression in order to switch genes on and off to do that job. It's also about the idea that normally cells are different because they've switched on and off overlapping but different sets of genes. So here's the figure, and it just talks about that. And basically it says we're going to take a cell from a host and take it from the host, and we are going to take that cell and we're going to put it in with the egg from another animal, a sheep in this case, because it was originally done in sheep. And so we've taken this totipotent nucleus from one cell in a body, put it in the egg cell of another, and then grow that up to have the surrogate mom give birth to a lamb that's genetically identical. The lamb is an identical twin with that sheep. Has the same DNA as that sheep. Like that. Okay, so that's the method. So now we're just talk. That's all. That's it. And we're just going to talk about that because in 2012, the Nobel Prize was given to two scientists. One of them was this guy, who was a British researcher, and his experiments were done about 50 years ago. So sometimes the Nobel Prize is awarded to somebody who's kind of done a lifetime of research, and that's what this guy, John Gurdon, had done. About 50 years ago, he did an experiment, and it was a cloning experiment. And in his case, it was with frogs. And so he took a cell from a tadpole, and he took the nucleus out. The nucleus has all the DNA in it. And he injected that into the egg of a donor cell, and he had already eliminated the nucleus of that egg cell. So he replaced the nucleus, eliminated the first one, and injected this one. He used frogs because they have really big eggs. So he could use a very fine needle and actually suck the nucleus from this cell into that needle and then inject it into that. And what he found is that the egg was able to develop into a tadpole like that. And so he took one cell from a tadpole and was able to generate a whole new tadpole. And he did more experiments. He took a cell from a growing frog and was able to generate a tadpole like that. And so he had a lot of success, and that was the first time really that people were showing good evidence of this concept of genomic equivalence. Whatever cell he took was successful, that each cell seemed to have all the genes, and that was big news at that time. And it's still news today. A big part of the last section of our course is learning about that idea, learning about that each of your cells has all the DNA. And then, before we jump on, oh, here's a slide I just pointed out. Here's early days, and this Wikipedia page, and it says John Gurdon attended Edinburgh and then Eton College, where he ranked last out of the 250 boys in his year group at biology, and was at the bottom set in every other science subject. Schoolmaster wrote a report stating, "I believe he has ideas about becoming a scientist. On his present showing, this is quite ridiculous." And so, not only does he have a great haircut, but he's a pretty epic character in science, and has contributed greatly, even though his initial showing was not that impressive in class. And that's a nice message for everybody, that you might not be the best to start with, but a lot of people hang in there and actually becomes a situation where they really contribute. So I said 2 people won the Nobel Prize. Here's the other scientist, Yamanaka in Japan. Gurdon in England did his major experiments 50 years ago. Yamanaka did his experiments only about 10 years ago. And these are the experiments we'll talk about. This is the modern science that takes advantage of some of the methods. And Yamanaka created what scientists refer to as induced pluripotent stem cells. So this is a story about stem cells, and people are really interested in stem cells, because stem cells can form lots of different cells. They can make many different cell types. And how can they do that? Well, they have the DNA, each cell has the DNA to do it, and somehow the stem cells retain the capability to do it. So here's Yamanaka's idea. So Yamanaka initially wonders, could we use the DNA microarray method that we just learned about in the previous mini lecture to compare gene expression in a stem cell with another cell type, really any other cell type, but let's look at this situation. So here's a stem cell, and when stem cells divide, they give rise to 2 cells, and the definition of a stem cell is that when the stem cell divides, it divides and forms another stem cell. And so very often you'll see this, that stem cells are self-renewing. So one key concept of stem cells is they have the capacity to form a variety of different cell types, and another is that they're self-renewing. So when this divides again, it'll make a stem cell. And so we have that happening in our bone marrow right now, or in our skin cells. And when the stem cells in our bone marrow and skin cells and elsewhere divide, they give rise to a stem cell and then this precursor cell. And this precursor cell goes on to be a blood cell in the bone marrow stem cells, like that. White blood cell, red blood cell, that's the precursor that's going to head down the pathway and become a specialized cell type. And our skin, it's going to become a skin cell, like that. So we got a precursor cell. And so Yamanaka just said, let's just compare by DNA microarray, let's compare these 2 cell types and ask now, how are they different? What gene expression is different between those 2? And if we can find genes that are expressed here and not here, why don't we study those genes? And what we can do with those genes is, so this is a stem cell gene, only expressed in stem cells. And how about if we take the gene and take off the promoter in the DNA for this gene, remove it, and replace it with another promoter that would be active in the precursor cell. In other words, what if we mix and match these 2 genes so that we can misexpress the stem cell gene in the precursor cell? What will happen? And Yamanaka found that if he misexpressed the stem cell genes in the precursor cell or any other cell, that cell will become a stem cell. So he was able to create a stem cell initially just from these precursors, but eventually they could take any cell from your body and turn it into what they refer to as an induced pluripotent stem cell. So now you've heard of embryonic stem cells, you've heard of adult stem cells, and these are artificially created stem cells, and scientists are very excited on because we can make a stem cell from your cells, and maybe that would help with some wound healing, regenerative medicine, things like that. So it's really exciting, and that's why these scientists won a Nobel Prize. It's really exciting. And it uses several of the concepts we've learned, an important concept that we've learned, this idea of misexpressing. So Yamanaka and other scientists found that they could misexpress just 4 genes in order to turn any cell into a stem cell, like that. And so they used initially DNA microarray, like we just learned about. So there's the stem cell, and there is a precursor. And basically they saw lots of different colors, but they saw several genes that were green. So these are genes that are expressed in stem cells, but not the precursors. They become very interesting genes, and they could misexpress those in the precursor in another experiment and see if they misexpress them, if the precursor cell would become a stem cell. And they were able to get it down to just 4 genes. If they misexpressed these 4 genes in a precursor or any other cells, they would create an induced pluripotent stem cell, a self-renewing stem cell. And that misexpression, I just tucked this in here because that we first learned about here, where this woman misexpresses PDGF by accident. So she misexpresses PDGF in the skin and in the location of this tumor, because by accident she has joined the transcription unit of PDGF with the promoter of a collagen gene that is normally expressed in the skin. And so now transcription vectors that bind normally to this promoter activate PDGF transcription. That gets transcribed into RNA and then translated into PDGF protein. So this idea of misexpression goes way back to this, accidental misexpression and unfortunate consequences, but then is used now by scientists, not by accident, but to intentionally misexpress genes. And so it goes back and this work ties into lots of stuff that we've learned about in the last couple of weeks. So here's a figure just from the web about take a normal cell, misexpress these 4 genes, create induced pluripotent stem cells, and then potentially produce any of the cell types that we're interested in your body. So let's say you have a problem with your pancreas, like maybe you're diabetic, maybe we could make some more fresh pancreatic cells and get them to make insulin for you, like that. Lots of potential. And these, I just want to talk for a couple more minutes. These are the genes that were misexpressed. And though they have odd names, but these are some of the several thousand genes, and they each code for a protein. And so we've learned about lots of proteins in our course, and these will be the last ones we mention, and their role. And so we learned about aquaporin, and we learned about LDL receptor, and we learned about RNA polymerase. You know, this list is actually quite long, each protein. And here's another set of proteins that are in our bodies, and they're orthologs in other species. They're very important genes in regulating stem cell development, like that. And we'll just look briefly at them, and we just go to their Wikipedia pages. So here's OCT4, and it says OCT4 is a protein transcription factor. Here is SOX2, and it says SOX2 is a transcription factor. Here's KLF4, and it says KLF4 is a transcription factor. So we just go back to that list. These guys are transcription factors, like that. MYC is a transcription factor as well. Okay? There's MYC transcription factor. So we remember what transcription factors do. Transcription factors bind to the promoters of target genes. So there's MYC or any of these others binding to the promoter of target genes and activating their expression. And it's not surprising if you said, hey, I've got 4 genes that can turn any cell into a stem cell. It's not surprising that the 4 genes code for transcription factors, because transcription factors can switch expression of lots of target genes. so they can switch on the various genes that need to be expressed in a stem cell. And then here's the last concept, and that is just, oh gosh, these people won a Nobel Prize. It seems like we have all our answers, at our fingertips. But when you stimulate a cell to be a stem cell, one thing you stimulate is you stimulate growth. And so right now, the induced pluripotent stem cells are great, but there's a lot of anxiety that in stimulating stem cell behavior and the growth of stem cells, you also might increase cancer. Here, MYC is being referred to as an oncogene, and the study of cancer is called oncology. A cancer physician is an oncologist, like that. So it turns out MYC is a strong stimulator of growth, and MYC is an oncogene. MYC is often mutated in folks that have cancer and the tumor, and is stimulating uncontrolled cell growth. And so it's exciting work that scientists are doing, and Gurdon and Yamanaka won the Nobel Prize a few years ago, and well deserved, and it really ties together lots of our course. Because we learned about the cell cycle, the regulated cell cycle. Here's a factor that regulates the cell cycle, MYC. And we learned about how MYC works. It's a transcription factor that's going to regulate the gene expression of target genes. And then we're learning that MYC is present in all genes, but it's only switched on in some. And some of the genes, some of the cells that switched on are stem cells. And the stem cells are fascinating cells, but they have the same genes as the rest of our body. They've just switched on a subset of the genes that are really interesting. And that's that. Cool. Cloning is awesome.